So let's just start by looking at this enzymatic pathway and see what's going on. So here we have inactive caspase, I guess that's how I pronounce it. I'm just going to roll with that. But it ends in ASE, so I know it's an enzyme. But if I didn't know that, like it's in the text already that it's an enzyme too, but knowing that helps you understand it, because there's just a lot going on. So it's in its inactive form, which means that the cell's always making this enzyme, but it has this little addition that makes sure that the enzyme's off and it's not working. But when there's a bacterial infection in step 1, this little portion of the inactive enzyme is cut off, and now this enzyme is active. So that's step 1. We have the in response to the intracellular pathogen, which is a fancy way of saying the bacteria is in the cell now. The inactive enzyme is cleaved and it forms the active caspase in step 1. So now that we have this active enzyme, it does two different things. First here, the first thing that it does here is here we have inactive interleukin. So it's also has a little addition to it, and it's in its off position here because it's inactive. So this enzyme will cleave off this portion and it'll turn it in its on or its active conformation. And if we look here, interleukin is a signal molecule that can activate the immune response. So now this active interleukin is a signal to activate immune response, which means it will like bring in like all of the phagocytes and B cells and all of those other cells of the immune system. So that makes sense because if you have a bacterial infection, you want to make a signal molecule so you can let the rest of the immune system know that we have some bacteria to kill. The third portion of it, so this N-terminal like gasturbin, like see here that this is its active form, but here this is the inactive. So what this enzyme will do is it will cut off the inactive part and it will make this N-terminal gasturbin, and this is the active protein. And it says in step 3, the N-terminal portions of several gasturbin proteins associate in the cell membrane and they form large nonspecific pores. So they form pores or holes in the cell membrane. So to recap, when the bacteria enter the cell, this activates an enzyme which will cause like the active form of a signal molecule that will signal the rest of the immune system, but it will also create this active form of these proteins that will form holes in the cell membrane. So now we're ready to kind of look at the question. We want to describe the effect of inhibiting step 3 on the formation of pores. So if we stop step 3, how will that affect pore formation and the release of interleukin? So if we stop step 3 and this does not occur, how will that affect pore formation? If we can't do step 3. Yeah, pores will not form. That's all you need to write. How will that affect, and then it says and, right? So that gives you 1 point, but here's your and. How will that affect the release of interleukin? How will it affect that, the release of active interleukin, if I stop 3? It won't. So all you have to write is that pores will no longer form. However, interleukin will still be released, and that's it. But that gives you 2 points on FRQ2, and that's one of the long FRQs with the big multiplier. So this is one that if it's question 1 or 2, it is worth your time to try to figure this out because of those bigger multipliers. Yep. And there will always be something in there that you don't really need to know stuff. It's just like reading a model, and then they'll get to stuff where you actually need some content. So here's where it starts to get into a little bit more of like understanding molecules and biology. So we need to make a claim about how cleaving inactive Kpase-1 results in the activation of Kpase-1. So we have to make a claim of how we go from the inactive Kpase to active Kpase-1. And what would you guys say? Like, how do you go from active to inactive? So by cutting off or cleaving the inactive Kpase, why would that make it active? Right, what's like the theme that we always need to talk about with molecules? Whether it's an enzyme or what did we changed its shape and now it can function. So by cutting off or cleaving the inactive Kpase, we changed the shape of the enzyme, so now it's in an active shape or an active conformation. So that's our theme, that structure always determines function. So if it was inactive here, it must have had the wrong shape, and now it's active, so it's in the active or the correct shape for the enzyme to work. Another way to say it, like you removed like a portion that, caused it, the domain that caused it to be repressed or something like that too. But that's just, you talk about shape and function, you're fine. Yeah. Like that's just a major theme of biology. Whenever you talk about something, like its structure is always related to its function. All right, so that's the first part of B where it says make a claim. The second part, a student claims that pre-infection production of the inactive precursor, so the inactive enzyme, shortens the response time of a cell to the bacterial infection. Provide one reason to support that student's claim. So what that student says is that if you have a lot of inactive enzymes, then you can respond to a bacterial infection faster. What's a reason that might make sense on why that might be true? A reason that would support that claim. This is a protein, right? What steps do I need to go through to make a protein? I say start where? My instructions for how to make a protein. What molecule has instructions for how to make a protein? Okay, so here's like the so structure is related to function is one big theme. Here's the other one. We go from DNA, and then we go to RNA, and then we go to our polypeptide for our protein, and then we see the trait. So this is like the chain of events. Why do I have brown eyes? I have a genotype that goes through transcription to make messenger RNA. And then we go through translation to make the protein that gives me the pigment molecule that's in my eye and you see brown. So linking DNA to a genetic trait is something we need to know how to do. Maitland, how you doing? You look like a light bulb just like went off. We okay here? Like, but this is a lot, right? Like, this took us like 6 weeks to like learn how all of this happened. It doesn't take the cell 6 weeks to make that molecule, but like if we have to go from DNA to RNA to make the protein to make this enzyme, that's going to take time. Or I could just make a whole bunch of it in its inactive form ahead of time, just in case. And as soon as I have a bacterial infection, we just cut the piece off and activate it. So, like I wrote it in a sentence form. My reasoning was cleaving a precursor molecule is faster than making a molecule via transcription and translation. Like you might not use the same terminology, but that's the reasoning that would support it. So that gives us the 2 parts of B. All right, in C,